This meeting is being recorded.
Thank you, Dr. Kurra, for your wonderful presentation. Thank you so much. Now the floor is open. Well, maybe the rooms are open for us to direct questions. In interest of time, we will only have 15 minutes allotted to questions. If you have a question, please raise your hand in the chat and we will have the host acknowledge your hand. Any questions for Dr. Kurra?
Dr.
Jennifer,
Bell.
yes, go ahead. Jennifer, while we wait for the questions, just want to thank you for giving me the opportunity to present this very important topic. I think getting the message out, and it's still very important to emphasize the need to vaccinate. We're still not completely out of this pandemic. We're reaching the end of it, but we are in a place where we are very fortunate to have an unending supply of vaccines. What we haven't seen is a 90% or higher uptake. And it's very important, especially given the luxury we have of getting it freely and widely accessible versus the rest of the world. And then we hopefully will get global equity in vaccination as well, because this virus is going to continue to evolve, to mutate, to form new variants of concern. And so I think it's very important. And thank you again for allowing me to present.
Thank you, sir. Thank you so much.
Mary Nelman had a question.
Yes, hello. Wonderful talk. That was very, very helpful. I end up, even though I'm retired, I get a lot of questions about vaccines. I saw a couple days ago, unfortunately, there was a news article of somebody who published in something called the Epoch Times, which is not a journal that I read, that the mRNA vaccine alters DNA in the liver, actually alters DNA. And the way the news thing was written is that the vaccine stimulates the nucleus to make DNA produce more mRNA, to generate more mRNA. As far as I know, this has not been peer reviewed or anything else, but it's getting a lot of legs. Even the study doesn't say that the virus intercalates into the DNA, but I don't know if you saw that or if you have any comments on it, because I'm going to get a lot of comments on it.
Mary, thank you for raising that. So this is based on a couple of studies that seem to suggest or indicate that it's possible mechanism. And the counter to that from the scientists, it's not from me, it's not from the lay public. Research clearly indicates there's a difference. So what we're actually talking, without getting too technical, is episomal DNA and episomal RNA. There's a difference in where the RNA is. So all protein is made in the cytoplasm. It's not made in the nucleus. And there will be copies of the DNA that are sent out into the cytoplasm, and as is copies of not the whole DNA, parts of it, in the form of messenger RNAs in order to make proteins for ourselves. So if you wanted to make protein, the blueprint still comes from the DNA, but the DNA itself does not come out of the nucleus, number one. And the number 2 point, which is where all these folks are very concerned, is the fact that there's a translation and therefore a possibility that there could be a two-way mechanism. But that fundamentally in all research has been showing to be not true, including the first paper that started this whole thing. Modified RNA is the reason why this RNA won't enter the nucleus. And this is why even the company Moderna is named after that. It says modified RNA. That's what Moderna stands for. And modified RNA has been over and over again. And this is why it took 10 years for Katalin Karikó, including losing her lab privileges, including losing her professorship. And she persevered and figured out to substitute a pseudo-uridine in place of a uridine in order to create this modified RNA. And modified RNAs just do the simple task of giving that possible for you to go and tell the cell to make a certain protein, and then it degrades and it has no ability to do anything more. And this is why that paper was groundbreaking. This is why 10 years, it took 10 years to get there, and then a further 15 years to create proof of concept and prototypes. And now, right now in the pipeline, there are several cures, ongoing trials, phase 2 trials right now, which is showing wildly successful results for neoplasms, including melanoma, which, as you all know, is deadly and most of the time leads to high rates of deaths. You know, this is going to continue to be debated, but the scientific community, the fundamental researchers understand this better than the lay public. And it's sad that this is being used and bandied around in order to drum up some fear when that is non-existent, at least from how well we understand this research.
Thank you. That's very helpful. I will talk to my relatives.
Yeah.
Tim Ridgway had his hand up, but it's down now. Tim,
I just had a brief comment, not a question, Dr. Kurra. As a gastroenterologist who mainly is doing administrative work now, I try to maintain a working knowledge of this entire situation. Your presentation reminded me of why I'm an internist. It was evidence-based, it was clear, and it pointed out all the faults. And in this day and age, when I've had discussions with pharmacists who tried to convince me that ivermectin should be utilized, and just seeing the science, and that's what internal medicine is, evidence-based, objective medicine. Thank you. I thought it was very well done and arms me with nice, concise information that I can utilize.
Tim, nice to hear from you. And you make a good point. You know, the pharmacist who's trying to convince you is based on what are very fundamental misconceptions. So when you say ivermectin prevents this virus, but the dose, the pharmacodynamics, those come into play, and then eventually you realize, yes, there are a lot of things that kill this virus. We can all drink bleach and we will get rid of this virus, but we can't drink bleach. We'll have severe, esophageal, as a gastroenterologist, you can tell me that right now. But thank
Very
you
bad,
again.
yes.
So one more question, Dr. Bill Fox.
So first of all, Dr. Kurra, thank you so much for that absolutely superb presentation. I was riveted, and I can't think of a better way to kick off the conference than that discussion. I think even the scientists who created these RNA vaccines were surprised at how great the initial efficacy was. And I'm wondering if you have any insight as to why these mRNA platform vaccines perform so well, say, compared to the traditional protein platform vaccines that we've had in the past. And then a second question, if I could sneak it in terms of looking at your crystal ball, when do you think we might have some idea as to whether we might require, say, annual boosters or boosters going forward?
Yeah, great questions. Thank you, Bill. The one thing about the mRNA is it is going to the fundamentals of how we make proteins. And I think that is the part, the surprise for the scientists about the success is based on the fact that, yes, we can make the cell make the right protein, the spike protein, and then the immune system goes into action. They were not expecting 80 to 90% in trials. And that is a new finding for us. And it's based on what I was talking about, conformational science, showing how the three-dimensional shape, every protein, its function is not based on the sequence of amino acids, which is what all proteins are made of, but it's the final three-dimensional configuration. So form is function when it comes to protein. And that research by Barney Graham, Jason McClellan, is the reason why this is 80% successful. But the fact that we're mimicking a way to make proteins that is foundational to all plants and animals, every plant and animal on this planet makes proteins through a messenger RNA. So that's one of the reasons why that's so successful. But the second part, the crystal ball, this is we're at the tail end of this pandemic. We've seen this act played out in 1918. It took 2.5 years before we were in the endemic stage. And endemicity is, if you look at how that happens, you change the immune landscape. So when this virus first broke out in December of 2019, it was truly a novel virus, and nobody had ever from an immune system standpoint seen a virus, a coronavirus, SARS-CoV-2, and so it was novel. It's no longer novel after you've had surges and vaccination. So the immune landscape today is 90% seroprevalence. If you look at the just the seroprevalence plus vaccination, we can say it's 90%. And when you get to that immune landscape, you will have some infections, but it will be endemic. It'll be like TB. We can talk about TB and SARS-CoV-2 in the same way. That's where we'll end up. So looking forward, summer of this year will be near normal, the new normal, and then we can see a small wave in winter, and it'll continue every winter. Fall and winter, if you look at the pattern, that's been the pattern the last 2 years when we've seen the largest surges.
Thank you, Dr. Kurra. Jennifer, are you still with us?
Yes, I'm still here. I was trying to reach our next speaker in anticipation of the time. Any further questions for Dr. Kurra?
I don't see
If
any.
there are no questions, I'd like to thank you, sir, very, very much for a very brilliant presentation and for being a good clinician and a supporter of clinical medicine and a supporter of clinicians as well. Thank you, sir.
Thank you.